All right, so before I introduce our guest, there are a couple things that are different in this lecture compared to the previous ones. So the title is Radiocarbon Dating for Conservators. The major difference is that throughout the lecture, Greg is going to stop periodically and let us do a little bit of Q&A, so that while he's going through the material, if you have questions, you can just put a question in the chat and we'll stop and answer it after 10 or 15 minutes or something like that, whenever Greg gets to a good stopping point. If you're feeling a little shy and you don't want to ask your question publicly, you can send me, Tiarna, or Nina a private message, and we can ask your question anonymously. So one thing that I want to stress for this particular talk is that we're very lucky to have Greg here because he has a really unique skill set in that he works with conservators, curators, archaeologists, art historians all the time. I certainly bug him all the time. And so he understands the parameters that we work within. So he understands how to, sample for a museum object. He understands, the kind of information and data that we're interested in. So there are a few different types of questions you can ask him. You can ask him anything about the material, but in particular, if there are practical questions that you have, like what kind of solvent might be useful to clean a drill bit before you sample, that's a great question. Or you can ask him why he likes aluminum foil so much. Those are good questions. So the other kind of category that you can look at are the theoretical things. So you can talk about radiocarbon dating oil paintings or mortars, lime plaster. Can you radiocarbon date silk, textiles, things like that? How would you go about that? So you can ask a broad range of questions, and we're very lucky in that he can answer almost all of them in vetting. If you have chemistry questions, nail them with them, because we can talk about beta decay, we can talk about how nitrogen goes to carbon, back to nitrogen, all that kind of stuff. So if you have questions, now's the time to ask them. And as always with these kinds of lectures, there are no dumb questions. If you think it's dumb, there's probably someone else who also has the same question. So feel free to ask it. And then there's Greg from a few years ago working in the radiocarbon lab in this picture here. And here he is in my studio in one of the instances where I was bugging him. So here he was helping me date this Egyptian coffin from about 2000 BC. So this is Dr. Greg Hodgins. He is the director of the University of Arizona Accelerator Mass Spectrometry Laboratory. He got his MSc in biochemistry from Cornell, and he got his PhD in archaeological science from the University of Oxford, where his dissertation was investigating methods of identifying pre-Renaissance artists' paints and glues. His research generally, I think most of his work is with geologists and climatologists and things like that. But he also works with objects of art and archaeology. So he works with conservators and historians and archaeologists. He has over 100 published papers where he's the author or co-author, which is ridiculous. Few of my favorites are identifying art forgeries by radiocarbon dating, dating gods, radiocarbon dates from the sanctuary of Zeus on Mount Lycaon, that's the birthplace of Zeus, and annual radiocarbon record indicates 16th century BCE date for the Thera eruption, that's Akrotiri, one of the most important sites ever discovered. So those are just a sample of some of his publications. So I'm not going to go on forever. Without further ado, I'm going to hand it over to Greg Hodgins. So Greg, whenever you're ready, you can go for it.
Right. Well, listen, thank you, everybody. I can't tell you how impressed I am that so many people are giving up their Friday nights for these lecture series. I mean, it's Friday, right? And it's Friday night. So I know probably many of you would just as well be sitting in front of the fire with your glass of wine, relaxing. And I'm hoping that we can relax anyway. Let me, I'm not sharing the screen here properly. Let's just,
so you should be able to click the green share screen button in the bottom there.
Got it. That's the one I'm looking for. Great. Good. Okay, start.
So on the top left, you should be able to just click from beginning and then we'll go full screen.
Great. Super. Okay, well, given it's Friday night and late for some of you guys, and as a sort of tool for attention, I'm going to start off by giving you a list of vocabulary words. And, I know it's sort of to help you pace out. I want you to listen for these words. And the deal is that you can only take a sip of your Nouveau Beaujolais when I say one of these words, all right? And I know that you, as a group, have been grappling with the seriousness of these seminars. So I'm giving an alternative list for the more abstemious of you. So those that want to sit gently, I have an alternative list. And then those that really want to go for broke, I have a third list, okay? So whenever you hear these words, you're allowed to take a drink, all right? So pick your list, charge your glass, and prick up your ears, and let's spend 45 minutes talking about radiocarbon dating. Okay, well, that's kind of, I'm going to talk about sort of 5 different topics, and most of it is trying to give you a foundation for just the basics of radiocarbon dating, and we can ask more sophisticated questions at the end, but I always feel like it's really important for people to just grasp the foundation. And once they've got that, then, you guys are all smart enough and comfortable with materials and chemistry that you can come up with, I'm sure, very intelligent questions. So I'm going to talk about the origin of C-14 dating and kind of dating theory, then kind of try to give you a picture of the nitty-gritty of sample preparation, instrumentation, and the process of measurement. We're going to talk about radiocarbon calibration, which is, to me, one of the most amazing aspects of the method. There's a variation called bomb spike radiocarbon dating, which works in the last half century at very high resolution. And then I'm going to talk about conservation materials and radiocarbon dating. So let's see how it goes. All right. We're in a difficult time, the pandemic, and there's a lot of things to worry about. But this should kind of put things in perspective. So, the Earth is traveling through space in actually quite a harsh environment. There's something called the cosmic ray background. And the cosmic ray background reflects the fact that in deep space, there are very energetic helium nuclei and hydrogen nuclei that I don't think the origins of those are actually known, but they have very high energies and very high velocities. And they're extremely damaging to organic molecules. And life on Earth wouldn't have evolved without an atmosphere and without a magnetosphere. So there's, in this particular diagram, oh dear, I forgot my, my mouse. In this particular diagram, yeah, this works. Yeah, there we are. Excellent. So here's the Earth, and it's this. The cosmic ray background isn't really shown here, but what you're seeing is not only is there the cosmic ray background, but there are a lot of very energetic particles that are coming from the sun, and you can see here that the Earth's magnetosphere is somehow shielding us from some of these energetic particles, but it's the Earth's atmosphere that actually shields us from many of them, and that's what actually allowed life to evolve on Earth. So I'm telling you all of this because carbon-14 is a class of radioactive isotope that's called a cosmogenic isotope. And that cosmos refers to the fact that there are stable gases in the Earth's atmosphere that are damaged by incoming cosmic rays or solar energetic particles. And those particles have enough energy that when they get to a thick enough portion of the atmosphere, the probability that they'll actually have a particle to nucleus collision is quite prevalent. And those collisions end up causing fragmentation of atmospheric gas atoms And then it's a series of reactions called spallation, and it's rather like, you know, shattering or movement of billiard balls, but collisions lead to other collisions. And this particular diagram is one that I love. It shows the Earth's surface down here, and then it's about cosmic ray production, and it shows the Eiffel Tower sitting down on the Earth's surface as scale, and then Mont Blanc, and then Mount Everest, and various other objects that sort of give you a sense of where these reactions are taking place. And the majority of these collision reactions are up in the stratosphere, but some collision reactions lead to subatomic particles that actually make it down to the surface of the Earth, and even some subterranean. So what's actually happening for the creation of carbon-14 is that things called thermal neutrons, which are released in the spallation reaction, collide with nitrogen-14, nitrogen nuclei. And so non-radioactive nitrogen has 7 protons and 7 neutrons. But what ends up happening is a proton in nitrogen gets converted to a neutron, and we end up with 6 protons and 8 neutrons in the nucleus. So that's a transmutation of nitrogen into carbon. All right. And of course, there are other gas molecules, and in fact, there are other cosmogenic nuclides that are created in this one. But because our atmosphere is 80% nitrogen, this is the most common reaction. And up in the stratosphere, this newly formed carbon oxidizes to carbon dioxide, and it joins the pool of non-radioactive carbon dioxide that's also a component of the atmosphere. And these reactions tend to happen more towards the poles because that's where the Earth's magnetic field is weakest. But really, carbon-14 is made in the atmosphere anywhere on Earth. And because we have weather that moves atmosphere and mixes it up so thoroughly, the concentration of C14 CO2 is pretty homogeneous no matter where you are on Earth, and whether you're up at high altitude or whether you're down at the surface of the Earth. So when CO2 gets down toward the surface of the Earth, and as I say, some of it has a C14 in it, plants do what plants do, which is they carry out photosynthesis. And that's the reaction where carbon dioxide is taken up into the leaves of plants, and it's incorporated into biomolecules to make sugars. And that reaction doesn't really distinguish non-radioactive carbon from radioactive carbon. And so that's where inorganic carbon becomes organic carbon, and that's the basic reaction of life. And so C14 tags along, and it then can subsequently be incorporated into any carbon-containing biomolecule. And of course, animals eat plants and make their biomolecules from plant biomolecules. And this reaction is actually quite fast. So the amount of C14 in the tissues of this animal and this tree and the atmosphere are pretty much the same. And because animals and plants are continually growing and they're replenishing the C14, this happens throughout life. But when living organisms die, if in the rare instance there's biological material that survives, the C14 concentration in that material drops. And so this is a scale showing the present, zero age, going back 6,000, 12,000, 18,000 years. And it's very crudely showing that the amount of radioactive carbon versus non-radioactive carbon falls off at a very predictable rate. And that rate is the half-life of carbon-14. And what's significant about using radioactive decay as a kind of clock is, of course, that radioactive decay is not influenced by environmental conditions at all. Temperature, humidity, pressure, light, all of those things, at least at the levels that we experience them, don't really affect these reactions that are going on in the nucleus. Okay. So carbon-14, as Luke alluded to, it actually has a very large number of applications. It's used as a tracer because it makes its way into all sorts of things. Of course, it's in the air, it's dissolved in ocean water, it's in ice. And then it's in all of this sort of long list of natural organic remains. And then it's also found in human artifacts and incorporated, I suppose, artificially, if you like. And so all of these things can be dated by the process of measuring C-14, understanding the rate of radioactive decay. So just to give you some very basics, of course, there are three natural isotopes of carbon-14, or of carbon, excuse me. Carbon-12, which is the most abundant, so that has 6 protons and 6 neutrons in its nucleus. 99% of carbon is carbon-12. 1% is carbon-13, so it has an extra neutron, but that extra neutron doesn't affect the stability of that nucleus. And really, the amount of carbon-12 and the amount of carbon-13 on Earth and you know, in our solar system reflects the proportions of those from the dust and gases that actually form the solar system. So in some sense, these are timeless. Carbon-14, of course, as I just said, is being created all the time, but it's very low abundance. So in modern materials, in the environment today and in your tissues, there are, we can kind of invert this, for you have to have 10 to the 12th, so 1,000 billion non-radioactive carbons for every carbon-14. All right, so and of course over time the amount of carbon-14 relative to these other isotopes drops. On that cartoon they use 6,000, 12,000, 18,000 because as an approximation of the half-life of radiocarbon, which is 5,730 years. So that statement of what's the half-life, it's a summary of the exponential decay of carbon-14, and it's the only number you really need to know, because exponential reactions, they're called first-order reactions, and they are only dependent upon the concentration of one component. The other thing I want to just tell you is, by convention, in dating, the 0 year for radiocarbon is 1950 AD, and it's very important that we have that convention. Otherwise, you can imagine if we used our contemporary year as the 0 age all the time, it would be very hard to interpret the literature. So we can call it the 0 year, or another way of expressing 1950 is to say that the material is 100% modern. In other words, it has as much C-14 in it as something from 1950 AD. And the useful time range for measurement, it's a little bit variable. It actually changes from day to day depending upon how the instrument runs. But we can measure roughly 10 half-lives, so somewhere out to 45 or 50,000 years to the present. And I guess the thing to realize is that when you're getting out to these very extreme ages, the amount of C-14 is extremely small, and it gets to a point where we can only quote ages as greater than 45,000 years, because it's a question you can't distinguish nothing that's 45,000 years old from nothing that's 1 million years old, if you like. Okay. So the first way that radioactivity was measured, and the way that Willard Libby, the inventor of the method, used it was to count radioactive decays. So we just described the creation of carbon-14, but it's unstable, and that's kind of the definition of what radioactive is. It's an uncomfortable combination of protons and neutrons, and at some point in time, the carbon-14 is going to rearrange, and it's actually going to go back to nitrogen, and it gives out this beta particle, which literally flies off into space. And when you use a Geiger counter, you're really using a detector that is detecting these subatomic particles, in this case electrons, that are the products of this nuclear decay reaction. Okay, so a modern sample that's 100% modern, 0 years old, the decay rate of those natural materials is 14 disintegrations per minute per gram of carbon. And then just by definition, something that's exactly 1 half-life old has half the disintegration rate per minute, and then 2 half-lives old, et cetera, on down. So this, you can sort of see that as you get older and older, the rate of decay of the organic material drops. And when decay counting is a perfectly acceptable method of measuring radiocarbon and using it for dating, it tends to require at least gram quantities of carbon. And it also, the older a sample is, you have to count for a long time. And kind of a rule of thumb for getting reasonably precise measurements is that you need to have at least 10,000 decays. And so you can just kind of do the math and see how long it would take to get to 10,000 at 14 decays per minute per gram of carbon. The method that we use is called accelerator mass spectrometry. And so this is not a decay-based method at all. It actually allows us to kind of partition the sample into its isotopic composition. So a modern sample, we literally quantify how much carbon 14, 13, and 12 is in the sample. And so, a modern sample has a carbon 14, 13 ratio that we'll just call X. And in reality, that X is 1 over 1,000 billion. But as a field, that's such a small number, it's cumbersome to even try to think in those terms. So again, we use this convention. It has a ratio of 100% modern. And again, 5,000, 1 half-life old sample has 50% modern, and 45,000-year-old sample has 0% modern. The advantage of accelerator mass spectrometry over decay counting is it requires a much smaller sample, 1,000 times smaller actually than decay counting. And this sample range kind of opens up the possibility for radiocarbon dating much smaller things, individual seeds, fragments of insects, individual purified compounds. So the sample size is reduced about 1,000, and really the measurement time is minutes rather than hours, days, or weeks. But the only disadvantage is it requires a big and expensive piece of kit. So I can show you. So I'm just going to leave the theory and talk about how we prepare samples. And so sample preparation, and then conversion of the carbon to graphite, then what the instrument's actually doing, and then we'll move on to calibration. So sample preparation is really, I think, one of the critical steps in radiocarbon dating, and we'll talk about why a little bit later, but it really is just a series of washing steps for the most part. And the archetypal radiocarbon sample preparation method is to make weak acid and base solutions, and these are mineral acids and bases, so they themselves don't contain carbon, which is critical. And really, we're just washing the samples with these materials. And really, the first step, this 0.1 normal HCl step, these can be varied basically in their concentration. But this is probably the most critical wash, and almost every radiocarbon sample goes through it. And that first acid wash, what's that? That is designed to dissolve mineral carbonates that are precipitated in a sample. So perhaps I should back up and just say that almost everything that comes to the laboratory comes out of a burial environment. It comes out of the ground for the most part. So whether it's archaeological or geological materials. And so those are organic remains that have presumably had groundwater percolating through them. They've been drying and wetting, and these carbonate precipitates that have from carbonates that have been dissolved in groundwater. Either it can be carbon from the air that's dissolving in groundwater, or it can be limestone carbonates that have dissolved in the groundwater. But as those enter and exit the sample, they can precipitate. And of course, they're depositing carbon that's not related to the actual organic carbon. So when you acidify any aqueous solution below pH 4.5, or pH 4, excuse me, carbonates, they just dissolve, and the CO2 bubbles off and leaves the system as a gas. So that's really one of the most critical steps. The next step is this Washington base. So we're inverting the pH of the solution to extremes, and there are classes of soil organic molecules, humic acids, fulvic acids, that are the breakdown products of older organic remains, and they're pretty much ubiquitous. And those humic acids are actually soluble in sodium hydroxide. So this is a way of washing soil organic matter out of the sample. The only disadvantage of this is that atmospheric carbon dioxide, it actually dissolves in basic solutions. So you have to do a final acid wash to bring the pH of the solutions back down to around pH 4 or 5, so that you're making sure that in the course of cleaning the sample, you're not recontaminating it with atmospheric CO2. And atmospheric CO2, of course, has today's levels of carbon-14 in them, so they can't be there. So actually, there's actually extensive water washing between each one of these steps, and it usually takes a couple of days. We do it at slightly elevated temperature, about 60 degrees C, but that's the standard. So then there are add-ons to these. Usually they're kind of preambles, and they're not so much used for samples that come straight out of the ground, unless there's a particular circumstance that requires them. But if there's the possibility that samples contain hydrophobic contaminants that may not so easily be released by these, we can do these additional solvent washes. And these are more used for museum objects or objects that have been handled extensively and may be contaminated with materials of a different age or different radiocarbon content in the sample, but are actually soluble in these hydrophobic solvents. And then the rest of the solvent series is really just designed to remove the organic solvents and return the whole system back into an aqueous system. And then some protocols actually use oxidizers, bleach or sodium peroxide or hydrogen peroxide. Whoop, sorry, back. And then the last kind of radiocarbon dating sample preparation is an area called compound-specific radiocarbon dating. So that uses chromatographic methods to purify a specific compounds from the target that you're trying to radiocarbon date. And the reason you want to do that is you want to have a compound that is unlikely to be free in the burial environment. So for example, with bone, hydroxyproline is in bone collagen, and it just doesn't really exist in the soil environment. So if you can get this and date it, you know that it came from the bone of interest. And there are other methods for these other kind of compound-specific targets. These require a lot of material, and it's very labor-intensive. And we don't actually do compound-specific dating in this laboratory, although I have done it in the past. So this is just kind of a photographic summary of kind of what cleaning looks like. So sort of unclean samples, and this may be a solvent clean sample, and then acid washed, and then this is kind of taken all the way through the ABA treatment. And you can see this color change. And what we really like is kind of white crystalline materials. And sort of here's another example. These are from an Egyptian sort of cartonnage. So it's a combination of textile and some kind of plaster, and there's papyrus there as well. And so you can see that these are the starting materials, and then these are the same sample, the same magnification after taking them through cleaning. And part of the point I want to make here is that sample losses during cleaning are substantial. So we are completely willing to sacrifice sample losses if we can recover something that's highly cleaned. But it's very difficult to know ahead of time how much sample you're going to actually lose. And I'm always asked, how big a sample do I actually need? And that's a very difficult question to answer, because you don't necessarily know how much you're going to lose. And sometimes you lose the entire sample, that These are both polymeric structures. Collagen is a polymeric structure. It's inherently insoluble, but of course, decay reactions can clip up those polymers and render fragments of them soluble. And so, you know, you throw out the collagen with the bathwater. All right. Okay. So the cleaning step is incredibly important, and the rest is just kind of, you know, bench chemistry. We, for solid organic materials, we extract the carbon out of them because we don't really care about all of the other elements, and it's just done by combustion. So you have an organic sample that has all the organic atoms in it. You add oxygen and energy, and you produce carbon dioxide and water, and then this array of other oxide gases that we don't really care about. But that's the extraction method. And then the CO2, we purify away from all of the other gases of combustion by a process called cryogenic distillation. And then purified CO2 is converted into solid graphite. So carbon dioxide it's hard to keep around the laboratory. We have, you know, glass vessels that we keep the gases in, and they have little O-rings in them, but over time there's diffusion across those. So we want to get the sample to graphite, which is a very inert material. It doesn't react with atmospheric compounds. It's stable in the lab. All right, so we measure the carbon-14, carbon-13, and carbon-12 composition of the graphite. And the goal, and it's actually very important, that the ratio of those isotopes in the organic remain is preserved through all of these steps. And you can do it incorrectly, and you can alter the isotope composition in any of these chemical reactions if you're not careful. So part of the skill of radiocarbon dating is ensuring that these reactions go to completion and you're preserving the isotopic composition. So this is a little tableau that kind of sums up everything. Here's a bone. It's actually a direwolf bone from La Brea tar pit. Here's some a drill site. This is the bone powder. It's a biomineral, so it's a combination of phosphate carbonate. So there's mineral carbon in there that we don't actually want, and collagen. So after we've removed the mineral and we've isolated the protein collagen by that acid-base acid reaction, you get this lovely white, fluffy, cottony sort of material. That gets combusted, and that's so we extract the CO2 as or the carbon as CO2 gas, and that's sort of represented by this gas vessel here. It's sealed at this end, got a valve at the other, and then the gas from there is converted to graphite, and that little smudge there is about 1 milligram of graphite, and that milligram of graphite is what's packed into this little holder here. So it's a bit like putting gunpowder into a cartridge, and so that we call those cathodes. That's what the sample looks like. All right, now to the kind of instrumentation. So the samples are measured in groups of 40. So here's a wheel here, and the measurements are made under high vacuum, and the instrument is developed out of the field of beam physics. And so one at a time, well, this is sort of closed into this sealed chamber. It's put under high vacuum. The samples are bombarded with cesium metal, which actually kind of knocks carbon atoms from the graphite, ionizes them, and if you want to think, sort of makes a vapor cloud of those things in front of them and gets them flying, if you like. And so this system here is called the ion source. And I just want to mention that there are 40 samples in this wheel. Every 6th sample around that wheel is a standard material called oxalic acid 1. It's the internationally agreed standard. All radiocarbon labs use it. All radiocarbon labs measure it because a C14, C13 ratio of that defines what modern is. So that's what allows laboratories to intercompare results with one another. It's a NIST standard, and I'm sure you guys are familiar with what NIST is. So it's the National Institutes of Standards. Over the course, we can put in one of these wheels a day, and it revolves around about 6 to 10 times over the course of 18 hours. And we end up collecting, of course, many, many measurements of those standards. We end up with a population of measurements of those standards. But we're also measuring the ratio of the unknowns that are in between each one of those. So the instrument's big. Rich was standing behind the ion source, which is at this end of the room. You can't get it into one photograph. This is actually an historic photograph. I picked it because that's Doug Donahue. Well, so here's the ion source here. It bends a corner here, moves through the accelerator tank. There's the edge of the tank there, and continues on around to the C-14 detector there. And that's Doug Donahue, who was the founder of this laboratory in 1980, and he just passed away 3 weeks ago at the age of 93. So it's hard to see the instrument through these photographs, but here it is in a plan view. So this is the ion source here. It's this long tube that has all of the atmosphere sucked out of it by high vacuum pumps. And we're generating a kind of cloud of negative carbon ions in here. And that cloud is drawn into a beam because if you have ions, you can manipulate them with electric fields and magnetic fields. And we want to draw that beam out and make it go down through this long path through this analyzing magnet here. And the C-14 actually comes all the way down to the end here. So I'm sure most of you are familiar with how mass spectrometry works, but the analogy that I use is how the fact that if you change the pathway of a mixed beam, components in it may change their direction differentially. And so, like, the analogy here is passing a beam of white light through a prism, and the different colors, the different wavelengths pass through the glass air interfaces at different angles, so you end up with this smear of the spectrum. For mass spectrometry, we have a beam of carbon that's actually a mixture of all of the isotopes. It doesn't pass through a prism, it passes through a magnetic field, which actually continuously causes the beams to bend, but in a completely understandable way. In a fixed magnetic field, the lightest isotope gets bent the most in the magnetic field, and it so ends up emerging at the greatest angle. And if this is a mixture of 3 isotopes, we end up with not a smear like you do with the electromagnetic spectrum, but 3 discrete beams. Each one is pure isotope. It's the pure mass. And the beams are, of course, discrete because there's no mass 12.5. There's no mass 13.25. You're separating those based on the different number of neutrons in the nucleus. And that actually happens. This is the analyzing magnet here. So we end up making a high velocity mixed beam of carbon that enters here. It's a very powerful magnetic field, and carbon-12 emerges from that with the tightest angle and bangs into a detector here towards the outside of this pie-shaped vacuum chamber. Carbon-13 makes a slightly less sharp angle, bangs into another detector that's slightly inward of the carbon-12 detector, and carbon-14 shoots down this path here, goes through another bend, and bangs into a detector here. So the isotopes are all ionized, so they're carrying an electric charge. So we don't really count them per se. We just measure the electric current that each one conducts. So, I mean, very simply, I told you that carbon-12 is 99% of carbon and carbon-13 is 1% of carbon. You know, if you had 2 light bulbs here and carbon-13 carbon was going through the instrument, you know, the carbon-12 light bulb would be 99 times brighter than the carbon-13. And the trick with what we do is that the current from this detector is, in modern materials, is 1,000 billion times weaker than the current from these detectors here. So the trick of this is reliably getting electrical signals from these detectors relatively. And in modern materials, the ratio is 1 part in 10 to the minus 12. but our detection limit is 1 part in 10 to the minus 15. So it's vanishingly small currents that we're detecting here. And we can do it with errors that are of the order of plus or minus 0.3% or 3 per mil. Okay. So fundamentally, what the instrument's doing, it's not measuring time, it's measuring the amount of carbon-14 relative to the amount of, in this case, carbon-13 in the sample. It could just as easily be carbon-12, but it's the ratio of the radioactive isotope compared to the non-radioactive one. That's the number. Okay. The last step is the calibration of radiocarbon measurements. And this cartoon here is sort of, I'm flipping it around from the Bambi cartoon. And so we've got the present here on the right and time unfolding as you move towards the left. All right. And this shows the graph sort of 1950 is where it starts. This black dotted line is trying to show you what a mathematical first order exponential decay equation looks like, okay? Superimposed on it is this blue line, and that's called the radiocarbon calibration curve. And that curve is actually depicting the amount of C14 in real samples of known age over time, over the last, in this case, 12,000 years. And the theoretical curve and the real curve overlap pretty well at first, but then they diverge over time. And this is important. And so I just want to stop actually for a second here. And let's see, stop share. Okay, any questions so far?
Yeah, we have a number of questions.
All right.
I'll take them as from the order they came in. So we had one from Susie, which was a great question. It was just, ice can be dated, ? So it was after you showing all the different materials that could be dated. So can ice cores be dated? I'm guessing is what that's related to.
Yeah, well, what you're actually dating is air trapped in bubbles within the ice cores. So it's atmospheric C14 is what you're dating. And you can imagine it takes large amounts of ice. But you know, the Greenland Ice Core project, that's exactly what they're doing. They're drilling down several miles into the ice core. They're carefully bringing up blocks of ice. They're melting them under vacuum and collecting the C14 or the carbon and doing C14 measurements.
Okay, and then the next one is from our buddy Arlen, and he asked, is there an interesting story about who discovered that carbon-14 exists? So not Libby who developed C14 dating, but who actually discovered 14C exists?
There is. Yeah, there is actually. That's worth a read. I'm not going to go into it now because it's sort of long and a bit of a digression. But yes, there is a very interesting story about the guy that discovered carbon-14.
I'll just note that it was Martin Kamen in 1940, and then Libby developed it in 47. And it is interesting. And Brooke asked, would love a little more detail about how samples are cleaned. Is it just baths? So maybe about how a sample is cleaned that is maybe contaminated with, you know, an adhesive or something like that. How is a sample like that cleaned?
Well, I mean, I think it's in some senses the dirty secret of carbon-14 dating, and no pun intended, that we go through the cleaning procedures and we assume that contaminants have been removed, because in reality, a single contaminant, truly characterizing it, understanding it, and understanding its solubility characteristics and proving that you've removed it from a sample is a master's thesis. And so it's actually just not practical to do that in most instances. So we go through these cleaning protocols, which are harsh and empirically shown to remove contaminants in many cases. And so we place our trust that those are sufficient. And I will say that that's kind of the allowable for samples that are coming from natural environments. we understand what soil organic material is like and how to remove it. I think the challenge for working on museum objects is the diversity of materials that they can be contaminated with. And they're, as you know better than I, often past treatments are not documented. The possibility of what could be there is enormous. And so that's one of the things that makes dating museum objects and treated objects so challenging. The reliability drops because we don't necessarily know if we've thoroughly removed the contaminants.
Yeah, so unfortunately, if you drill into an area for a sample and you accidentally drilled into some epoxy that you didn't know was there, and then you go to clean the sample, the pre-treatment clean is not necessarily going to get rid of that.
Absolutely right. Yep, that's right.
Okay, and then so we had another one from Arlen asking, why don't you want the phosphate carbonate from bone when you're getting a sample?
Well, the main reason is that biomineral can seed the precipitation of mineral carbonates from groundwater. So you often, so it can be quite challenging if you, for example, pull bone out of the ground and you target the bone carbonate, you will get carbon out of it, but it's very possible that the radiocarbon concentration of that will be a combination of the biomineral carbon and groundwater carbon.
We had an anonymous question asking, why not just analyze the CO2 gas rather than graphite? So why not just look at that CO2? Why go through all that process of converting it to graphite and using, those iron catalysts and things like that to get that graphite?
It's a great question. And in fact, there are some laboratories, there's new instrumentation that allows you to directly inject CO2 into the ion source. It's really just about the efficiency of making the beam. If you have a graphite in there, it's a solid sample, and when cesium bangs into the solid sample, it yields a lot of carbon atoms. If you're spritzing a gas into that ion source, the gas is just diffuse and it's harder to get a strong beam of carbon from it. But it is being done now, and it's actually very useful for extremely small samples.
And then one last question was from Nina saying, did I understand correctly that nitrogen is actually the origin of C-14?
You did, yes, that's exactly right. Yeah, so rather nuclear reactions or decay reactions are transmutation reactions. So the alchemistry of, lead into gold. A nuclear decay reaction, the elemental identity of these things change. And so carbon comes from nitrogen and it actually returns back to nitrogen.
I think a lot of us, especially in conservation, we get through organic chemistry and then we go and celebrate. And so when we start talking about protons and neutrons and beta decay and these reactions that occur that are actually very important for natural biomolecules, it starts to get to be a little overwhelming. How does a proton turn to a neutron? Does a neutron turn to a proton? So I think that's really confusing for a lot of people. So you're not alone in that one, Nina.
Yeah. Well, and so I'll use that as the next step, which is, I think you learn about photosynthesis starting probably in 8th grade or 9th grade. And, I heard it, yeah. But I really was not awed by the reaction of photosynthesis until I got into carbon-14. And we started with the cartoon of Bambi and the tree. So we know that C-14 goes into plants and becomes part of plant organic material. I want to actually talk for a second about wood, because just to remind you guys that this is a section of a tree that from Mount Lemmon. It was harvested a couple of years ago. It produces annual rings. But the amazing thing about a tree is that the only tissue that's alive here is this thin layer of cells called the cambium layer between the outermost wood and the bark. And I have this sort of desire. You got to work with me on this. Okay, so I'd love to have these special glasses that would make the hard part of tree, the dead part, invisible. So you could walk through a forest wearing these glasses And what you would see are these beautiful diaphanous columns of transparent silvery living cells going up into the sky and branching out. And the ends of those, the twigs, would be little tiny silvery gossamer tubes that would be ending in leaves. And of course, the fluids are moving up and down that layer of living cells. All the inside that you can't see, it's dead, it's inert. So these would be like, giant inverted jellyfish. And the photosynthesis is happening in the leaves. That's where the carbon dioxide is being incorporated into sugars, small molecules first that are mobile. And of course, those are coursing down through the trunk out to the roots, and the things are moving in both directions. And the cells that are here in the trunk that aren't photosynthesizing, they're pulling sugars out of that stream to feed themselves for their own metabolic activity. And over the growing season, they're dividing, right? And they're making daughter cells, and the daughter cells are kind of pushed to the outside, and the mother cells are crowded into the back, and they go through a kind of tree cell menopause and finish out their life by taking the sugars out of that stream and making cell wall material. And then they die. And what's absolutely spectacular to me about this, of course, is if it's a tree that is making annual rings, then each one of these cellulose, the dead cellulose in each one of these rings, is an archive of that year's atmospheric C-14 content. It's absolutely magnificent. And so this tree we know is felled in 2018, right? And the rings are annual, so we can count back and we can know the year that every ring was actually formed. And the radiocarbon community, what they've done is they've cut out these rings and measured the C-14 concentration of them. So that's the link between atmospheric C-14 levels and time. And the other point that's really important is that everything that's alive on Earth at the same time has the same amount of C-14 in it. So once we've gone to a tree and we've reconstructed how much C-14 was in the atmosphere in the past, things that were alive in those past years have the same amount of C-14 in them. So this is the basis of the radiocarbon calibration curve, okay? So that's the kind of starting point. And then the next bit I find just awesome. And that is that, okay, what am I doing here? Share. Hey, went back to the beginning. What happened? All right.
Yeah, I think you could click whatever slide you were on and then do from current slide.
I'll do that.
There were some more questions that came in while you were talking, but we could save those for the end.
I don't want to go over time. I feel like I'm kind of lagging a little bit here.
No, I think you're okay. I'm personally fascinated, so I can listen to you talk for hours, so I'm personally fine.
Okay, so from current slide, there we are. All right, so I told you that this blue curve is the calibration curve, and it's the record of what the atmospheric C-14 levels were in the past. And it's based on measurement of tree rings. And I'm showing you a curve here that starts in 1950, and in this particular slide, it goes back 12,000 calendar years. We now have that curve extended out to nearly 14,000 years. And it's all made of measurements on tree rings. And the way it's constructed is using the science of dendrochronology, which was actually invented at the University of Arizona at the end of the 19th, early 20th century. And it is a way of actually linking the ring count between living trees and dead trees back in time. And it has to do with the fact that trees that grow in the same regional environment share the same random pattern of good and bad growing years. And that translates into shared patterns of fat and thin rings. And so I just told you we cut this tree down in 2018. There's a pattern of fat and thin rings during the beginning of this tree's life that a tree that died, but its life overlapped with this tree, would have that same random pattern of fat and thin rings towards the outside of its growth. So you can count the rings back and assign them to particular calendar years from your tree that you've just cut down, and you can transfer that ring count to a nearby dead tree, absolutely, and continue on counting down so you know when each one of those rings formed. And then you transfer that count from the inner rings of that tree to a nearby older tree. And the dendrochronologists have spent 60 or 70 years piecing these together, and they have assembled these amazing ring sequences that go back, as I say, 14,000 years. And the radiocarbon community has done what I just mentioned, which is cut them out and document how much C14 each one of these things hold. So kind of completing the loop, we don't do mathematical calculations to assign ages to things. Based on the fact that everything that's alive at the same time on Earth has the same amount of C14, we get an unknown sample, we measure the amount of C14 in it, and we just go to this curve and we query it and say, okay, well, our sample had 60% of modern carbon levels in it. Which tree rings have the same amount? And we identify the cluster of tree rings from our library of previous measurements. We find which ones are the same, and we know the age of those tree rings by ring counting. So we assign that age to our sample, and that's it. Okay? It's really, I mean, that's the whole talk in a sense. If you get that, it's pretty awesome. And so there are squiggles in this curve though, and the squiggles turn out to be really important. So I'm just going to magnify one there. And they have a huge bearing on the way actual radiocarbon dates are translated into calendar time frames. All right. And you know, revealing these wiggles and squiggles through this process of dendrochronology and making measurements in trees has actually told us about the process of C14 generation. It's actually reflecting the fact that the sun is this very dynamic object that is sort of at various times throwing out more or less of these particles. Its magnetic field fluctuates over time. The Earth's magnetic field fluctuates over time. So these fluctuations all influence how many cosmic rays enter the Earth's upper atmosphere and how much C14 is produced. So there's small variations. So what it means is that, you know, here's a particular time here. We've measured the amount of C14 in trees from whatever this is, 310 AD. The trees that are older than that actually have more C14 in them than our tree here for a very brief period of time, reflecting the fact that in previous decades, more C14 was being made in the upper atmosphere for whatever reason, okay? So that there's this general trend that the older something gets, the less C14 it has in it. But when you get into the weeds, these little oscillations are really important. So, so I just described to you how we take a radiocarbon measurement and convert it into a calendar age range there. And this is a graphic representation of exactly that. We call it a calibration plot. So the y-axis shows the amount of C14, and this is what our machines tell us. What's the amount of C14 in a sample? And our measurements are represented here as a bell-shaped curve, all right? It's represented as a bell-shaped curve because we actually make multiple measurements. We get a statistical sample, and they're not all the same. And it's just in the nature of many types of measurement that that's true. And the belief is the more times you measure it, if you average them, the mean value of that average will approach what the real value is. So this is a population of measurements that we made. The mean value represents our measurement here, which is 94.4% modern carbon. But the distribution of measurements on either side of that mean is a reflection of how well the instrument ran. Some of those were higher than the mean, and some of them were lower than the mean. And the farther you get away from the mean, the fewer measurements have those extreme values. And a very low precision measurement might have the same mean value, so it would be a very speared out bell, and a much higher precision measurement might have the same mean value but be very narrow. So radiocarbon dates are never really expressed as a single number. They're always expressed as ranges, reflecting the fact that true measurement has variation in it. But so that's the measurement. As I say, we just translate it across to the calibration curve, which is these are these previous library of measurements on tree rings of known age, and then we translate it down to the calendar date scale. But we don't actually use the mean value. We really just use the range of values that our multiple measurements represent. And traditionally or conventionally, we quote our ages at the level of plus or minus 2 standard deviations from the mean. And when you quote a range that's 2 standard deviations from the mean, by definition, you're quoting 95% of your measurements. So there's a 95% chance in this measurement that the true value, the true radiocarbon content of the sample falls somewhere between these two bounds. And those translate into a range of ages that go, in this case, from 1410 AD to 1475 AD. Okay, and some people use the plus or minus 1 standard deviation from the mean, which is narrower. It's the width at about half the height of a bell-shaped curve. So you may be tempted to go for this 68% plus or minus 1 standard deviation range, which is indeed narrower. But if you quote that, there's actually a 1 in 3 chance that the true age of your sample falls outside of that. So this bracket here kind of represents what the range of plus or minus 2 standard deviations, and this is plus or minus 1 standard deviation. So I picked this part of the curve because it's relatively straight, and you can see that the bell-shaped curve here, which is mathematically symmetrical, is almost reproduced on the time axis there. So the most probable age is actually right here, and it kind of represents the most probable value of radiocarbon content of the sample. But so that's that measurement there. But of course, if you strike other areas of the calibration curve, strange things start to happen. So, and it's just the fact that this particular measurement strikes the calibration curve here. In other words, there's a cluster of tree rings of this age that have the same amount of C-14 as a sample, but there's another cluster of tree rings right here that also have that same amount. So we end up with this bimodal distribution of possible ages. And we can't say which one of these is correct. I mean, our sample really did just come from one of them, but with the radiocarbon measurement alone, we can't identify which one it is. And then at other times, I mean, when it's a real disaster, and this is a very famous section of the calibration curve from 800 BC to 400 BC, it's a period when the C-14 concentration of organic materials in the environment worldwide hardly changed for 400 years. So if you're trying to date anything that was alive, even just for the briefest period in here, the only answer you're going to get is this enormous time window of almost 400 years. Okay? So again, our measurement, the measurement that's depicted here, has the same precision as the previous measurements. It's just its location on the calibration curve actually determines how broad the calibrated age range is. All right? And then I think, I'm assuming that most of you actually are working on museum objects, and just for the, I think the majority of objects are kind of within the last 1,000 years, and radiocarbon dating within the last 600 years is particularly challenging. So this is the calibration curve from 1400 to 2000. It's actually ending here at 1950, which is the 0 year for radiocarbon. But there were these periods of very rapid change and oscillations in atmospheric C-14 concentration. So kind of like the example I just showed you, if something was really from 1500 AD and you did a radiocarbon measurement on it, the answer that you get back would be, 1450 to 1650. And if something was really from 1600 AD, you get pretty much exactly the same result again. And so that's kind of clustering around this part of the calibration curve. And then things that are more recent, within the last 300 years, they have slight variations in them, but they still have these atrociously large date ranges. So within the last 500 years, really, radiocarbon dates give you two answers: 1450 to 1650 and 1650 to 1950. So it's pretty disappointing in many respects, and it's probably why C-14 has a limited number of uses. And I don't want it to be completely dark, but there are techniques and there are methods that can take those atrocious calibrations and make them more useful. And I think you're an audience that really appreciates that part of what you're doing is telling the stories of objects. And doing that is very challenging given the incomplete information you have to work with, the challenges of the analytical data that is provided. You often have to try to tell a story from multiple pieces of not very good information. And carbon-14 is another one of those instances, at least in a lot of cases, where it's not nailing the answer, but it's giving you a piece of information that you can integrate in with other types of information. And that's what I'm trying to illustrate here. So this is a calibration plot of a New Guinean wooden statue. It had a pretty atrocious series of possible date ranges here, but it had provenance data. So the piece was collected before 1910, although the radiocarbon date indicated the possibility that its true age was in this range, 1910 to 1950. We can exclude that because we know the piece was collected before then. And there might be a situation where the piece was collected in a geographical location that the people that made the piece didn't occupy that location in previous centuries. So information like that could exclude this. So you can, by combining external information, take a kind of atrocious radiocarbon measurement and maybe make it useful. And the one thing that is important here is that these are three discrete peaks saying how old the piece could possibly be. But you have to also recognize that these valleys in here are saying there's no probability that the object could be from these time periods. So radiocarbon dates in this time can be useful for hypothesis testing. If you have a hypothesis that this object really was from 1700 AD and you make a radiocarbon measurement like this, this is not consistent with your hypothesis. Okay, so I don't know if it's trickery, but I think it's sort of giving you an idea that you can use these measurements in a sophisticated manner to take what isn't the best information and actually make it useful. All right, so the next topic is really using radiocarbon in the post-nuclear age. So we've looked at the calibration curve already. It ends in 1950. There's a kind of reason it ends in 1950. And that's because, so here's the time scale now. There's 1950. We're kind of looking back. And I've de-emphasized those oscillations and just sort of said there isn't actually a lot of change over time in the radiocarbon content of things. From starting in 1955, the C-14 concentration of organic materials on Earth skyrockets to nearly double the amount. That happens between 1955 and 1963. And since then, it's been dropping back down to almost natural levels. And that is a consequence of carbon-14 produced from above ground nuclear tests. So the natural process, of course, is cosmic ray particles, these high energy helium and hydrogen particles with high velocities entering from space. But around the mushroom cloud, there are also high energy particles leaving the mushroom cloud, and they collide with nitrogen in the atmosphere around the cloud and end up making C-14. And between 1945 and 1963, there were literally hundreds of above ground nuclear tests. There were enough of them that they almost doubled the natural level of C-14 in the environment. So that's a really important tool because it clearly distinguishes, post 1955 modern from
natural levels of C14. And we use that bomb carbon marker in all sorts of ways. It's used in the food industry, narcotics testing. We use it in forensic science for high resolution dating of human remains. And of course, it's used in art authentication. So, if you have a work that is pre-1955 and you make a measurement in it and it shows the bomb carbon, it's a pretty clear indication that something is up. And I've used the term forgeries here. This really, it's a very blunt instrument or a blunt terminology, and I'm a little bit embarrassed using that for this audience. I should have, this was for another audience, but here is a calibration plot using the bomb spike, and so you have a 19th century canvas. You measure the amount of C14 in the canvas. There's the measurement of C14, and this is the atmospheric C14 levels over the past 65 years. So our measurement intersects with this brief period in the 1950s when it skyrocketed from natural levels upward. And then there's another time period here on the way back down, as I didn't mention, I should have mentioned, 1963 was a moratorium on above ground nuclear testing worldwide. And so the nuclear nations stopped doing that. They went underground and underwater. So they stopped this contamination. And the C14 levels in the atmosphere are coming back down, not because of radioactive decay, but because atmospheric carbon dioxide dissolves in oceans. And the oceans are basically just sucking that bomb-derived carbon out. And oceans are vast and lumbering, so that pulse of bomb carbon has now gone down into the deep oceans, and it'll be bubbling back up for thousands of years into the future, but it's essentially removed it from the terrestrial environment. So, so here's a result here that is a pretty clear indication that this is not a 19th century canvas, okay? But with the caveat that picks up from our previous discussion in the question period about cleaning samples. We can only really conclude that that's absolutely true if we're sure we've really cleaned the canvas. And that is the kind of segue into the very last bit of the talk. And I can stop here. Do you want me to stop for questions, or 4 more minutes?
Let's go through the contaminants, and then we'll do questions at the end.
Okay. So I think I actually covered this a little bit. That this is a stratigraphic section, and we're looking at an artifact that came buried from somewhere in there. While it was sitting there and presumably not moving, organic materials from the surface and from past surfaces, decay products have been percolating down through it. Water has been picking up organic remains of different ages and percolating it up past. So this thing has been absorbing organic materials of different ages throughout the time of its burial. But we understand the chemistry of what those natural organic materials are, and we know how to clean them out of the sample. When you start getting into artifacts, the range of possible contaminants just explodes. So, there are natural materials that could be applied to artifacts, animal glues, egg and milk-based glues, starches, celluloses, waxes, varnishes, resins, but also, of course, natural oils from handling and passing around, and for funerary and temple objects, unguents that have been used to anoint these things are part of their use life. And these materials may have different radiocarbon concentrations than the object themselves. And so residues of them, if they're present, if they're younger and they're not removed, they'll artificially shift the measured radiocarbon date younger. And the other side of this thing is that with the 20th and 21st century, there are many varnishes, adhesives that are they're petroleum derived. So petroleum products are, of course, organic materials They had C-14 in them at one point in time, but they're 360 million years old. So the C-14 is long since decayed away. But petroleum is the basic skeleton for the synthesis of all kinds of molecules that are in the present day environment and used in the restoration and conservation and creation of works of art. And getting back to this thing about cleaning, we know how to remove natural organic molecules that come from the burial environment, but a lot of these things are designed to polymerize in situ and not be soluble and be durable. And so our simplified cleaning systems may not necessarily remove them. And so you just have to be much more cautious about making statements that you understand what's going on when you try to radiocarbon date conserved or restored or handled museum objects. And the last thing is just sort of quickly talking about dating fine art. And it's just sort of a similar story, I think, that fine art paintings, if you like, there are it's pigments, of course, dissolved in some kind of a medium, adhered to some kind of a substrate. The pigments themselves, many have not carbon in them, but several actually do. Of course, you can have carbon black, you can have copper carbonates in the green pigments. White pigments can be chalk, as can be the grounds in paintings. So there's complexity in there, and of course there's carbon in the binding medium, and there's carbon in the varnishes that go on the surface of paintings. And then there's the complexity of the layered structure of these things on the surface of a painting. And the layers can be very thin, and they can be pretty challenging to date. So I'm mentioning this because the radiocarbon community is getting to the point where it's starting to target individual layers within paintings using gas ion sources that allow you to sample extremely small samples of material and make measurements on them. So we're heading towards this pretty sophisticated time, but you got to temper it with what I talked to you about dating within the last 500 years. There can be tremendous technical achievements in dating these small samples, but if you're working on paintings that are actually from the last 500 years, you're nevertheless going to really get these broad radiocarbon dates on them that are 1450 to 1650 or 1650 to 1950. So there's work to be done, I think. So that's it. You guys have been very patient. We've talked about C-14 dating in theory, how we make the samples, the instruments work, and radiocarbon calibration, which is just awesome. And then bomb spike dating, which allows you to exploit the fact that C-14 levels in the environment over the last 65 years have been dramatically perturbed, and they've changed so rapidly we can make very high resolution measurements of the C-14 concentration of objects and assign very narrow dates, plus or minus a year or two. And then I just touched on conservation materials. So with that, I'm going to end it and take whatever questions. Thank you so much for your attention. I hope you found it interesting, and I hope you might find it useful. I'm available to talk to anyone in the future if you have particular projects you want to talk about.
Well, I think I speak for everyone when I say that was incredible. Thank you so much. That was absolutely phenomenal. So a silent round of applause. That was amazing.
You're so welcome.
So we have a ton of questions, and I think rightly so, because this is just so interesting. And I think people can probably see why you and I are friends, because we both love trees in the same way. And you mentioned that conception of trees to me years ago, and I've never looked at a forest the same way. So we had a question from John saying, I recently saw something about benchtop radiocarbon dating. Any comments on that? So actual benchtop instruments for radiocarbon dating.
Yeah, well, they are, they're getting much smaller. And so our instrument is from the year 2000, and I think it was about $3 million at that time. There are instruments that are almost benchtop, certainly that would almost fit in my office, which is, I don't know, 8 feet wide by 12 feet. And
And those are much simpler. They have the same precision as our instrument, and they're about $3 million. So it's still a pretty heavy investment. But the number of radiocarbon labs is increasing around the world, and there's literally millions of radiocarbon dates have been made over time, and there's certainly upwards of 100,000 measurements are made a year worldwide. So I think in my talk I've talked about problems, but I don't want to overemphasize that. The method really does work well, and it's been tested a lot. So, but I want to give you guys a sophisticated sense of what the challenges might be, and hopefully arm you with a little bit of knowledge to think about how you might use it, and whether or not it would be useful to you in your work.
Yeah, I think we all tend to focus on the negatives of whatever our technique or practice is. So I don't think anyone blames you for that. So we had a question from Julie asking, so is there an analogous calibration for marine calcareous sediments?
Yeah, well, yes. And I'm really grateful that you asked that question, because the tree ring calibration curve goes out about 14,000 years. And it's made from trees that they grow in northern climes where there's a real winter and a real summer. So those are the ones where you reliably get annual rings. If you move more towards the equator, trees may have seasonal growth patterns, but the rings might be tied to pulses of rain. They may not necessarily be annual, and you might not be able to see them. But so it's Irish oak, German oak forests that provide a lot of these trees. And beyond 14,000 years, those things were covered by continental glaciers. So that's the limit for tree-based calibration curve. Beyond that, people are looking for other types of annual records, and some of them are actually marine. Some corals actually grow with annual rings visible in their calcification. There are a few areas of the world where sediments are laminated. So there'll be a dry season where dust and sand will blow off the land and settle to the bottom of the ocean. And then there'll be a wet season where organic materials from the terrestrial environment will be washed out into the ocean, and it'll be deposited. And this sort of reliable dry, wet season alternating sand and organic materials, they're called varved sediments. And those things are also, people are going into those varved sediments and measuring the radiocarbon concentration of those organic layers and counting them. So we have a varved sediment record that goes back almost 40,000 years now. It's not quite as reliable as tree rings, and I didn't really mention it just for the audience, but we do have a calibration curve that goes back much farther, and there are terrestrial and marine calibration curves, because the radiocarbon concentration of the ocean is different than the terrestrial environment. So you need to know what you're dating, and kind of from the standpoint of museum objects, we often get asked about to date ivory, and here's my little ivory object. So the question that I always ask is that terrestrial ivory or is it marine ivory? Because the radiocarbon concentration of the ocean is the equivalent of about 500 years older than it is in a terrestrial environment. So you really need to know the animal origin of ivory, whether it's narwhal or whale, or whether it's elephant or whatever, in order to do radiocarbon dating on it.
Mm-hmm. Yeah, I think looking at the marine aspect really gets into how deep radiocarbon science is, and how the oceans churn C-14. It's really incredible. So we had a question from Susie that was 4 questions, but it's kind of getting at the same one. And so the questions are, how many trees have been dated from different regions from the Earth? So, do we have dates from trees from every region? For example, Mediterranean, maybe Chinese forests, things like that. Is there no variation in that radiocarbon at all? So for example, if you were looking at a 35 AD sample, would it be in the same of all of the trees around the Earth? Would it be the same carbon content, essentially?
So there are dendrochronologists use, it's a dating technique, and it's much more precise than radiocarbon is. You know, if you go into an ancient building and find a beam, and you have a dendrochronology sequence from that particular regions that shows a pattern of fat and thin rings, and you look in your building beam and you reproduce that series of fat and thin rings, you can assign the cutting date of that beam to a single year. So the dendrochronological dating is vast. The thing that makes it different than radiocarbon dating is it has to be regional. So you're building these chronologies based on forests that have shared the same kind of sequence of good and bad growing years. So there are these tree ring sequences from all over the world, but the radiocarbon community doesn't use a lot of them. But to get to the essence of your question, there are slight differences, regional differences, and it has to do with the way atmosphere circulates. Atmosphere actually doesn't cross the equator particularly easily. Atmospheric circulation happens to go around the Earth in kind of bands, and there are 3 of these bands within the northern hemisphere and 3 of them in the southern hemisphere. And there's less land in the southern hemisphere. There's more ocean in the southern hemisphere. And I told you that carbon dioxide dissolves in the oceans. There's exchange between carbon dioxide in the oceans and the atmosphere. So that the southern hemisphere has slightly lower levels of C-14 than the northern hemisphere. And then there are all these sort of weird things like volcanoes often release CO2. It's basically melted limestone from deep in the earth that has no C-14 in it. And there are these vents of CO2 that are coming up, and they can locally perturb the amount of C-14 in vegetation around them. People have done studies down the boot of Italy. There's very tectonically active regions down Italy, and they've showed locally that vegetation C-14 is depressed.
Yeah, that's been one of my favorite recent papers, was the one that you and Charlotte and all the folks at the Laboratory of Tree Ring Research did, looking at Thera and looking at the the calcium depletion in tree rings from the eruption of the volcano at Thera.
Yeah.
So Jessica asked one of the questions I was hoping someone would ask, and she asked, can you talk about best practices for taking a sample from an object, specifically from a wooden artifact? So I think I can also throw in, why do you love aluminum foil so much as part?
Yeah, thank you for that question. I'm a little bit embarrassed I didn't cover it more thoroughly. Well, to start off, so the question of how do you sample a wooden object? If it's a sculpture, I think I had a picture of a sculpture, in one of my slides. So, of course, before you make a sculpture, the first thing you've got to do is cut down the piece of wood. And the date, so that's the human act that you actually want to date. But it's the radiocarbon date of the outermost ring of that piece of wood, just under the bark. That's the cutting date of the wood. When you make a sculpture, of course, you're actually carving away that information. And imagine this was much larger, and say its life was 1,000 years. I cut down this thing today, and I made one little sculpture out of the outermost wood, and say it had a range of 500 years or 400 years of data of wood age in it. And we radiocarbon dated that. If we weren't careful, we would conclude that object was 400 years old. If we made another object from the center, that wood, is 1,000 to 200 years old. And if we weren't careful, we didn't understand what we were doing, we would assign the age to that object, 800 or 1,000 years old. But it's not. It was, made from wood that I just cut down. We've lost that information. So wooden objects have this inbuilt age, and you've got to be very careful about what the conclusions that you make from a radiocarbon sample from wood. So the best practice, tree specialists often pay attention to wood that has this heartwood versus the sapwood. And many tree species, or some tree species, actually have a kind of defined proportion of heartwood and sapwood. So maybe you're working on an artifact that was carved from the center, but you've got a little nick of sapwood in it. You go to a tree specialist and say, okay, well, I've got a little bit of the sapwood. How many rings do you think were carved away from there? So you end up with these sort of estimates of how much inbuilt age there was in the piece of wood. But yeah, so that's kind of risks, I suppose. If you have the option and you want to drill into a wooden sample, if you can drill into the end grain so that your bit is actually picking up just a few rings, then it's probably a better radiocarbon sample than going down this way, where the drill bit would be grinding out wood from a whole series of different rings, material of different ages. Okay, so that's kind of sampling wood. And the last thing I'll mention with wood is that there is a technique called wiggle matching. This tree, if you measure the radiocarbon concentration at many points along it, you'd actually be recreating a little bit of the calibration curve. And that little chunk of the calibration curve can actually be used to date this with very high resolution. I'll leave that behind. But why do I love aluminum foil? So aluminum foil, you can go to the grocery store and you can buy a roll and you can peel it off. And if you don't put your fingerprints all over the center of it, it's carbon free, it's lint free, it dissipates static, it's ubiquitously available. You can take a sample, fold it up into your piece of foil, seal it in, put it inside a little Ziploc bag, and it has a barrier. It's a barrier to stop contamination. So I can't tell you how many times I receive, little vials of samples that are stuffed with cotton to sort of stop them from rattling around, or contaminated. The samples themselves have been contaminated by the packing. It just happens over and over and over again. The one weak thing about aluminum foil is if your sample is damp and maybe acidic, if it stays in the aluminum foil for weeks, months, or years, it will actually corrode the aluminum foil, and your storage system will fall apart. We have 40-year archive of samples in the basement, and you can see that the foil wasn't robust in a lot of those situations. But certainly for you guys collecting a sample and sending it to a radiocarbon laboratory, foil is a really, really good option.
It's rare to find someone who loves aluminum foil as much as they love trees, and we've got the guy right in front of us. So Helen had a question, and I think she got some bad advice. She wanted to date some wood that was thought to be 250 years old, but she was told that it couldn't be done because of the 1945 atomic explosions in the atmosphere. So what are your thoughts on that? I would imagine that if we had something from 1780 or so, or 1770, something like that, we should be able to get a pretty decent date on that.
Well, okay, so I just told you about wiggle matching. So if you just took a single sample from that piece of wood, it would fall into that. It would give you a radiocarbon date of 1650 to 1950, because that single sample's radiocarbon content would fall somewhere in that horrible series of oscillations. But if I don't know what the object is, but if you're in a very lucky situation where you can see a range of rings and you can take multiple samples across that, maybe 3 or 4 samples across that, because you can count the number of rings between your sampling locations, you know how much time that range of samples represents. And so the radiocarbon concentration between those samples will vary according to the atmospheric levels that existed during its lifetime. And so you can reconstruct a little chunk of the calibration curve using your sample itself. And so I told you that there are these horrific oscillations within the last 400 years. But, if your sample allows you to reconstruct a little chunk of that oscillation, in other words, as the tree grows grows, if you're getting increasing amounts of C14 in every sample, there's only a limited number of positions on the calibration curve where atmospheric C14 was increasing. And if you're in a situation where the C14 levels are increasing and then decreasing across your sample, then that will date this wooden sample with really high resolution in a way that you just can't do with materials you have to take a single sample from. Okay.
Mhm.
That's hard to describe. Yeah, but I appreciate the people shaking their head, giving me the idea that they understand what's going on.
Well, it's a difficult region to date within, We had a question from Katie who asked, can you use the ±1 standard deviation if something is less old and therefore the calibration curve is tighter? And related to what you just talked about, can you repeat during which years you said it was a period of 400 years, the 14C didn't change very much?
I'll answer the second one first. It's 800 BC to 400 BC, and it's pretty much the worst plateau of the Holocene. It has various names, but it's called the Hallstatt Plateau for a lot of Europeans. So, you can quote whatever time range you want, whatever uncertainty you want. I think the thing to be clear is that when you quote ±1 standard deviation, you can only really use that terminology referring to the amount of C14 in the sample, because our measurement of the amount of C14 is normally distributed. It's symmetric. We make those measurements, there's differences, we average them, and we describe the differences by this normal curve, okay? As soon as you do the calibration, the curve that you generate of calendar years is no longer symmetrical. It's not normally distributed. So you can't really talk in terms of a mean and standard deviation of calendar dates. It just doesn't work. Imagine that calibration across the Hallstatt Plateau. We want to summarize these things. We want to be able to write in a paragraph what the date is. And in fact, oftentimes, you end up quoting these extreme values that are just the earliest possible date and the latest possible date. But when you do that, you give up something. And what you're giving up is the insights that some bracketed time ranges are completely impossible. So the graphic representation of a radiocarbon date gives a lot more information than just the written description. But of course, sometimes that's all you can do is a written description.
Okay, and then we had a question from Nina asking, is there also a marker with cesium during this time as well? And I think it was during when you were talking about the nuclear testing.
Yeah, no, yes, there is. I guess, wow, so I think the thing that makes carbon-14 so valuable is it's not a contaminant. It's at the very core of what the organic materials are. There are dating methods that sort of exploit the appearance and disappearance of contaminants within the system. But cesium is not a biological molecule. But you can use sensitive instrumentation. There's a whole field called dendrochemistry, which does exactly that. It looks for traces of environmental contaminants within tree rings and uses the fact that you can date the tree rings to date the appearance and disappearance of contaminants. Okay.
Okay, and then we had another question asking, how much of a minimum sample by weight is required to carbon date in case of a fabric like silk?
Oh, well, so silk is challenging to clean. That's really the difficulty. So it's a polymer like collagen, or actually like cellulose, but it's actually quite fragile. So you have to use very weak acids and very weak bases to clean it, because when the pH of those goes to extremes, the polymer breaks up and you just end up dissolving the silk. So we end up making these very gentle cleaning solutions to try and accomplish that. But of course, the weaker your solution, the less sure you are it's clean, the less kind of confidence you have in the validity of the radiocarbon date. And I think that brings out a really important point, that we characterize uncertainty mathematically. That's what the date range is sort of expressing. There are some types of uncertainty that are very easy to actually give a real error estimation for. But there's a lot of other uncertainties that arise that are very, very hard to quantify. And these sort of uncertainties that arise from the question of how clean is the sample is a perfect example of that. It's very, very difficult to quantify that uncertainty, and yet it's there. And you know, I don't know that there's a great solution or a way to communicate that. But you know, when I write reports about, for example, dating silk, I will bring these things up, and you know, it's sort of like I feel sometimes I'm shooting myself in the foot. I'm saying, hey, here's a radiocarbon date, but you shouldn't trust it. But that's the reality, or you should temper your trust in this radiocarbon date for these reasons, because we don't know this about that sample.
So we have a lot more questions. I'm going to throw one of my own in though, because I think it's an interesting one. When you're taking a sample, say from wood, what diameter drill bit do you use? How do you clean the drill bit before you sample? And how much does it cost to get a sample of wood analyzed?
Sure, all good questions. So, you know, it's mass that we want, and when I drill into a wood sample, I assume that there's contamination going in from the outside. The diameter of the drill bit, it sort of depends on the size of what I'm working on, but I want to minimize damage, so I tend to go for small drill bits. Usually an 1/8 or 3/16 is the maximum. And I also look at the width of the tree rings. So if they're very tiny, I might go down to a 1/16. And you know, you can drill in a long way, and so a small diameter drill bit can yield a lot of material if you go in a long way. So it's the drillings that we want, and we don't want dust. We want a very sharp drill bit with a lot of pressure and slow revs, so that it's kind of shaving the material, because it's much easier to handle shavings than it is to handle dust. And we'll get fresh drill bits. We'll clean them with acetone and alcohol, and then distilled water, and dry them very thoroughly. And then I drill in a short distance if it's an old artifact and I'm looking for a color change in the drillings. Throw those out, and then go back into the same hole again and drill the sample. And you know, if you can possibly manage it so that you have a little square of tinfoil, my favorite material, that will capture the drillings as they come out of the hole, and they'll fall into the center of your piece of tinfoil, you're off to the races. I mean, many times the geometry doesn't allow that to happen, and it can be very challenging. But you know, we want maybe 10 to 20 milligrams of material. That's usually enough if the wood's in good condition. And the cost is, it's somewhere around $400 a sample. It's a little bit cheaper if you don't need the extra solvent washings, but our prices are pretty close to our at cost prices. So, I mean, we're a university. We're actually, you know, a service to help scholars carry out their work. We do private stuff, and we charge private companies or individuals double what we charge everybody else. But it's just a very small portion of what we do.
Yeah, I want to emphasize that literally anybody here, you can send samples to Greg's lab, and there's a phenomenal staff there that can clean them for you. So you don't have all of the stuff that he talked about cleaning. All of the technicians there, who are phenomenal, take care of that for you. So if you take your sample, you literally just send it to them, of course wrapped in tinfoil, and then they will date it for you and send you a report. Arlen had a really cool question, and it's something I'm interested in. I don't know if it's something we can go into here off the cuff, but he said, I'd like to hear about how the math works when analyzing wiggle dating results on wood. I don't know if you're feeling up to that or not, but if you are, I'd love to hear it too.
Yeah. So the calibrations that I've shown you, this is kind of important. It's freely available, and I'm not. There's a lot of great statistics and Bayesian analysis that's behind those calibration plots. But even at my level, I just call up the program, I enter our radiocarbon measurement and its uncertainty and the name of the sample, and I press a button and the calibration plot spills back out. Okay, so that's the simplicity of calibrating a date. There are, I think, 4 calibration programs that are freely available from around the world. I use one called OxCal, which was developed by Oxford University. And there are more sophisticated levels of analysis that are built into these programs. And these more sophisticated programs use Bayesian statistical methods. And that's a mouthful, but Bayesian methods really just allow you to take information sort of from outside of your measurements, but that are pertinent to your measurements, and incorporate them in to narrow the probability of your radiocarbon dates. And they allow you to do that in a statistical manner, so you can actually probabilistically revise your dates to be narrower. Now, let me try to put that into words in a more simplistic way. Remember, I showed you that stratigraphic section where there was the dog skull beside a stratigraphic section. Imagine you had a series of charcoal samples throughout that section. Okay, the stratigraphic layering of that section tells you that there's an age sequence of those samples. The bottom one is the oldest, the top one is the youngest, and the 2 in between have sequentially related ages, assuming that the stratigraphy has that integrity, right? But if it has that integrity, then the interrelationship of the radiocarbon measurements on those must fulfill the physical orientation of those. So the calibrations of each one of those may be quite broad, but the fact that they have an age relationship based on their position places constraints upon what the age might be. All right? So Bayesian methods are incredibly powerful. The exact same thing is true with a tree and wiggle matching and tree ring measurements. These rings are older than these ones. There's no question about it. And that places constraints on what the calibrated age might be. They might actually have calibrated ages that are virtually overlapping, but you know that this is older than this one. And so that the statistical analysis of that will modify the probabilities of those ages to reflect that. Okay, does that help, Arlen?
Let's see, where's Arlen at? We got a double thumbs up on that. So I think that
cool. So I think, what's the math? These programs, they're not idiot proof, but there's a way to enter the data into those things. You select a kind of sequence analysis. You can describe the sequence, and you can say, okay, these related objects are completely discrete in time, or you can say there's a possibility they're overlapping in time. You can put boundaries on them, and they'll give you slightly different answers. But yeah, so it's actually incredibly valuable. And in the history of radiocarbon, sort of the first thing was Willard Libby discovering the method. The next thing was probably accelerator mass spectrometry. The next revolution was calibration and correcting radiocarbon dates to known levels of C-14 in the past. And the fourth revolution is really the Bayesian statistical analysis of radiocarbon dates. And I haven't touched on it here, but it's enormously powerful for, in some senses, taking really broad bad radiocarbon dates and generating in a probabilistic way credible explanations and credible time ranges for them. But just like sort of all computer programming, you know, you're taking that external information, like the stratigraphic relationship of those, you have to assume that the stratigraphy actually reflects this unfolding of time in a rational manner. So there's all kinds of examples of stratigraphy where layers are inverted and mixed up, and you know, so if that assumption that you made is wrong, then the output is garbage. So, you know,
yeah. I think a lot of us that have also gone, you know, delved into physical chemistry and quantum mechanics and all that. After a while, you realize everything is statistics and probability. So it's just another place that's popping up. Amy asked, can we date cremated bone? Which I think is a great question, because I think a lot of people ask me in particular, can I date charcoal? Can I date, you know, burned wood from an archaeological site? Can I date human remains that have been burned? Things like that.
Yeah. The answer is yes, and it's actually a really interesting alternate story to everything we've talked about with C-14 making its way into organic remains. The mineral structure of bone, as we talked about, is kind of open and it can exchange with the environment. And so we tend not to want to measure the C-14 content from buried bone, okay? Because the carbon might not just come from the bone. When you cremate bone, the crystal structure of the bone reorganizes. And in fact, there are a lot of tiny crystals in biomineral. And when bone is cremated, those crystals become much larger and much more highly ordered. So what was a kind of open, spongy system becomes a closed system. And the natural carbon in bone during the cremation process burns out. Most of it leaves. But when you're burning it, there's fuel there, and that's burning wood or whatever was used to make the cremation fire. It turns out that the carbon dioxide from the wood makes its way into the cremated bone. And then as the crystal structure locks down, it sort of fixes the fuel C-14 level into the bone. So when you're dating cremated bone, you're not dating when the animal or the person lived, you're basically dating the age of the cremation fire. And they're just there's some great science where they discovered that they thought they could get reliable dates from cremated bone, and then working its way through experimentally where the carbon actually comes from. But yeah, it's a well accepted method.
Okay, so we have an anonymous question saying, I have a radiocarbon dating chart from an early 17th century painting. I'd be interested to see what Greg's take on it is. Can I screen share it for a moment or two? Oh, okay. Do you want me to?
Calibration plot? You have a calibration plot from a painting date?
I could give you a, let me see, I'll give the person co-host abilities. If you'd like to share your screen, you could share it. That sounds very interesting. And I think that actually kind of wraps up our questions for the evening. So unless anyone has any last minute questions, I think we might give Greg a break now.
I'm fine. I can't thank you guys enough for your attentiveness, and it's such a pleasure to speak to people that know and understand objects and have a sophisticated appreciation for the methods that are used to try to help them tell their stories. And so I've always enjoyed my collaborations with Conservators, and I hope that I'll be able to collaborate with some of you in the future. So thanks so much for listening.
Greg, I just wanted to let you know, I don't know if you have been monitoring the chat at all because you've been talking a ton, but I just want to let you know that before you sign off, sort of scroll through the chat because you're getting a lot of love and it'll make you feel good. And I also wanted to really thank you for being able to take the cosmic view and share with us your knowledge. It was really fascinating.
I'm glad you liked it. It was my pleasure.
I'll send you the chat, Greg. People are really appreciative of this. I know I'm appreciative of this. I learned a lot. Every time I talk to you, I do. So thank you very much for this. Thank you everyone for showing up for the talk. We all really appreciate it. I hope everyone enjoyed this and learned a little bit about radiocarbon dating and how you might be able to incorporate this into your practice, whether you're in an institution or a private conservator. It's something that's available to everyone, and it's a really powerful tool. I know we talked a lot about the drawbacks of radiocarbon dating, but it is an extraordinary science in itself, and an extraordinary technique that we can utilize. So thank you again, everyone, for showing up. Thank you, Greg, so much for that. That was absolutely phenomenal. So we'll see you all at the next event we'll announce soon.
Bye, Chris.
All right. Thank you, everybody. Take care.